Okay, so photosystem 2 then is, as far as what you need to know about the photosystems, just kind of know their basic structure. So yes, there are 10 transmembrane protein units that help move electrons around, and there are 2 specific P680 chlorophyll molecules. Really, the key there is that the P680 chlorophyll molecules make photosystem 2 P680. There's high oxidation atoms in manganese there in photosystem 2, which is why they can pull electrons away from water. And of course, your electron transport chain is directly associated with it, because that is the pump that then moves the hydrogen into the thylakoid space. Photosystem 1, very similar to photosystem 2, but they have P700 chlorophyll molecules instead of P680. Photosystem 1 gets its replacement electrons from the electron transport chain, because they lose their electrons. Once photosystem 1's electrons get re-energized and passed off to NADP, the replacement electron for photosystem 1 comes from the electron transport chain. And so here you see an image of the complete process, the photosystem 2 passing its energized electrons onto the electron transport chain. The electron transport chain utilizes, so here's my electron transport chain. It's utilizing the energy of these electrons to pump hydrogen into the thylakoid space. There is a low concentration out in the stroma, and it builds a high concentration gradient inside here. That electron, once it gets passed through the ETC, has no energy. It gets re-energized at photosystem 1 and then passed off to NADP, forming NADPH. These hydrogens that build up in here, right, that creates that electrochemical gradient that we saw in cell respiration, they will go back out of the thylakoid, back to the stroma through the ATP synthase, creating ATP. Now, so chemiosmosis then works just like it did in cell respiration, and by chemiosmosis, right, we mean that electrochemical gradient has been built up, and those protons will move through the ATP synthase then to create ATP. You know, those hydrogen ions, or those protons, can only exit the thylakoid through the ATP synthase, and we use that exergonic flow of hydrogen to phosphorylate ADP. Now, if we need additional ATP made, because the Calvin cycle is not a one-to-one process, it doesn't require one NADPH for every one ATP. It actually takes a little bit more energy. If that is the case, then some electrons from photosystem 1, when they get energized, instead of going straight off to NADP, they'll actually detour back through the electron transport chain, creating more of a chemical gradient. So that's why we call it cyclic photophosphorylation, because that electron cycles back through the ETC so that it can make a little bit more ATP. And then from there, eventually that electron will go out to NADPH, to form NADPH. So that takes us to the Calvin cycle. So the light-dependent reactions take place on the thylakoid using the photosystems and chemiosmosis to make ATP and NADPH. The Calvin cycle then is actually where we're building carbohydrates, and we use the energy from our ATP, and we use the reduction potential found in the hydrogens on NADP. Calvin cycle, named after who discovered it. There are other pathways, photosynthetic pathways. So this one is called C3 photosynthesis. C3 for the type of organic molecule that's created in the Calvin cycle that eventually becomes glucose. The key to the Calvin cycle is the fixation of carbon dioxide to RUBP. Because at this point, carbon dioxide is a gas that's dissolved in the fluid stroma, and until that gas joins with or is accepted by RUBP, we can't make any sugar. So Rubisco is an enzyme that is also in the stroma along with RUBP, and it has an affinity for RUBP and carbon dioxide, and it helps join those two together to complete the first step of the Calvin cycle, which is carbon fixation, because there are 3 steps. So carbon fixation is when RUBP and carbon dioxide join together, forming something called PGA, or phosphoglyceric acid, or phosphoglyceraldehyde. Second phase is what we call reduction, and we know that reduction means you are adding an electron or a hydrogen to something. So PGA here gets reduced using the hydrogen from NADPH into a molecule called G3P. From that point, G3P can either become glucose, or we can do the third step, not or, it's both of those things actually. We use it to make sugar, and G3P is used to regenerate RUBP. And as far as, how many turns and how many carbons, clearly if you're making a molecule that its formula is C6H12O6, then the cycle is going to have to incorporate 6 carbon dioxides, and it incorporates really 1 per turn. So 3 turns of the cycle bring in enough carbon to make a single G3P. 6 turns of it produce enough carbon, fixed carbon, to make a single glucose molecule. So here you see kind of a breakdown of the process. Carbon fixation at the top here, where the enzyme Rubisco takes RUBP and combines it to CO2, forming PGA. I am not concerned about these numbers. Don't worry about we have 12 molecules of that and 10 of this. Just know the major intermediates of the process. So here we have PGA. PGA gets phosphorylated into biphosphoglycerate, then it gets reduced into G3P. Okay? Once G3P is produced, it will either become glucose, and it takes 2 G3Ps to become a glucose, or it's going to go back here to be converted or regenerated into RUBP. So the output then of the Calvin cycle, it's not really about making glucose, it's about making G3P. G3P is the simplest sugar molecule we have. It's a 3-carbon sugar, and we do use it to make glucose and sucrose. And actually, sucrose is how we move sugar around in the plant. We don't move glucose around, but we combine glucose and fructose to make a sucrose, and then that is what gets stored in the plant. Eventually, it will also get stored as starch as well. So, as far as energy goes, right, we understand the big picture here between photosynthesis, and respiration, for example. So, photosynthesis uses what respiration made, right? Respiration produced CO2 and water. And then in photosynthesis, those are the reactants of photosynthesis, while the products of photosynthesis are what respiration starts with, right? Glucose and oxygen. So the two are definitely linked. There is evidence that, really that glycolytic, that really very, very basic ancient glycolytic pathway is kind of the reverse of the production of glucose from G3P. So there's a lot of similarities. We also see similarities in the electron transport and ATP production, right, in the two. So again, that whole endosymbiont theory, how those two original organelles, the chloroplast and the mitochondria, could have been related, could have been a symbiotic relationship in a complex molecule, but then evolved into separate pathways, is kind of evident by all the similarities that they have. Right, and so this image does a nice job of showing the similarities between the two.